Inviting me, and this looks like it's going to be a great symposium. So for decades there's been considerable debate over the role of selection in species divergence. So on one side you have the staunch neutralists who believe that the vast majority, if not most, substitutions that occur during evolution are neutral or perhaps maybe deleterious. But on the other side, you have the tenacious selectionists who believe that a substantial fraction of substitutions that occur have been driven by positive selection. And so perhaps if you'll indulge me for a minute, do we have any neutralists in the audience? Oh, okay, okay. And selectionists. Wow. I thought no one was going to raise their hands. Well, regardless of one's views, the neutral theory provides a well-defined null hypothesis and a statistical framework for testing that hypothesis that can now be overlaid on top of the massive amounts of genome sequence data that is currently being collected in a diverse array of organisms. And so one of the most commonly used statistical tests for neutrality is the Macdonald Kreitman test, which very simply compares DNA polymorphism within one or in some cases two species to divergence between species. And the test is elegant in its simplicity. And so the test compares the number of non-synonymous and synonymous SNPs within species, the ratio of those data, to the number of non-synonymous and synonymous differences between species. And if one assumes that synonymous sites are neutral or perhaps at some equilibrium with very weak selection, then the expectation is that this ratio of non-synonymous to synonymous variation within species should be equal to that of between species. And so conceptually, this equality is fairly easy to understand, which is if a certain proportion of non-synonymous mutations are neutral and allowed to become polymorphic within a population, the same fraction should be allowed to become fixed between species. Okay, so there's two deviations that can be detected. And for detecting these, this is just done with a chi-squared or some sort of contingency table test. And one is in the case of positive selection, if some number of non-synonymous mutations are under positive selection, then they should rapidly become fixed between species, and this will increase the number of non-synonymous differences between species above what you would expect just for neutral non-synonymous changes. Because adaptive substitutions occur quickly, they're not going to contribute much to non-synonymous polymorphism within species. So the end result is that positive selection is going to elevate the ratio of non-synonymous to synonymous variation between species compared to within species. You can also have deleterious mutations, which will become polymorphic in a population, and that will increase the amount of non-synonymous variation within species. But these won't become fixed for the most part. And so deleterious mutations are expected to increase this ratio of variation within species compared to between species. Of course, in many organisms, probably deleterious and adaptive substitutions, deleterious mutations and advantageous mutations occur. So both are going on. And a number of methods have been, and so because both are going on, both polymorphism is elevated and divergence is elevated, but there's been a number of methods that have been developed to simultaneously account for both deleterious mutations and adaptive substitutions. And the way that, one of the ways in which this can be done is based on the fact that deleterious mutations have a tendency to be kept at low frequencies in the population. And so deleterious mutations can be detected by comparing rare variation to common variation, with rare variation being this non-synonymous to synonymous ratio being elevated above common variation due to the presence of deleterious variants at low frequencies in the population. And then positive selection will increase the ratio of non-synonymous to positive selection can be detected by comparing divergence to common polymorphism, which should not have very many deleterious variants. And so the McDonnell Kreitman test can be used to estimate the fraction of non-synonymous changes between species that have been fixed by positive selection. And the way in which this is done is by comparing the degree to which the non-synonymous to synonymous ratio of divergence is elevated above that more than that of polymorphism. So in this case, it's about 2 times higher, leading to an estimate that nearly 50% of non-synonymous changes have been fixed by positive selection. So this elevation is assumed that everything that causes this is due to positive selection, greater than what you'd expect from the numbers for polymorphism. And this can be done for a single gene or across the whole genome. And this has now been done in numerous different species. And so this is just a summary of 40 different species that the McDonnell Kreitman test has been applied to, either with whole genome data or data where a large enough set of genes have been examined that you can have some sort of estimate of the genome-wide estimate of positive selection. And so this includes vertebrates, plants, insects, fungi, and bacteria. And what's fairly easy to see is that there's a good deal of heterogeneity where many species show an estimate of positive selection of 0, which means that those two ratios are either not significantly different from one another, or the N-S ratio of polymorphism is actually greater than the divergence. And then a whole bunch of other species where the ratio of non-synonymous to synonymous divergence is much greater than polymorphism. So what can explain this heterogeneity? I think there's a couple of simple explanations. One is, study design, the methodology used, sample size, and all these things. And while this could have a slight impact, I don't think it's believed that these are really causing the differences that I showed you on the previous slide. Another possibility is that the frequency of positive selection could be different in different species. And then a third possibility is that there's some problems with accounting for the effects of deleterious mutations. So in some species this might be accounted for well, in other species it might be more problematic. So one thing that we know is going to, that we know can be important is the population size, or the effective population size of a species. And the reason is that species with large population size are going to more effectively fix adaptive substitutions, and at the same time, they're going to more effectively remove deleterious mutations. So species with large population size should have a stronger signature of adaptive evolution. And there's actually some nice data supporting the role of population size. This is from a review paper where you can see a nice relationship between population size and alpha, which is the fraction of non-synonymous changes estimated to have been fixed by positive selection. However, there are some species that are outliers, one of which is the budding yeast Saccharomyces cerevisiae. So estimates of the effective population size in yeast are in the order of about 10 million. If you plot that on this graph, you get a fairly obvious outlier in that it has a very large effective population size, but it's from a number of different studies, the estimated fraction of adaptive substitutions is 0. So another way of looking at this data is to just look at synonymous variation within each species, and then synonymous divergence between species for all of those 40 species to which the McDonnell Kreitman test has been applied. And the species in red are the ones that show evidence of positive selection. And the ones in black show no evidence of positive selection using the McDonnell Kreitman test. And what you can see is that there is some relationship here. There's a pretty strong tendency for species with evidence of selection to have high rates of polymorphism, and also some perhaps a bit slighter tendency for them to also have lower rates of synonymous divergence. And in this context, yeast is no longer an outlier. The symbols for yeast are these stars, and they no longer appear to be an outlier. And so, if population size is not the explanation, then what is? And I think this is a difficult question to answer. Being at a conference on recombination and evolution, I think it is worth pointing out that Hill-Robertson interference between selected sites is very well could have an impact, and this is going to depend on a number of selected sites in the genome. Species with more variable sites in the genome are probably going to have more selected sites, but at this point it's difficult to tell whether that is the explanation for this pattern. So today I wanted to talk about potential explanations for the lack of evidence of positive selection in budding yeast Saccharomyces cerevisiae. So, sure. These stars right here. So one of the first things to know about polymorphism in cerevisiae is that there's a very strong population structure. So this is a tree generated from 37 yeast genomes that were sequenced at low coverage, and the gray shading highlights strains that all share a number of SNPs that are unique to those strains. So a large number of SNPs that make these strains different from all of the other strains. And so there's, of course, very high FST and strong structure. There's also indications that quite a few strains show admixture between these groups, which is not easy to see on this tree, but if you run population structure analysis, there are some strains that are mixtures of these different groups. And so one of the things that has been quite interesting in this study by Alishev, is to look at the ratio of non-synonymous to synonymous variation across these different groups. And what's observed is that within each of these subpopulations, you find the highest ratio of non-synonymous to synonymous variation. And then a lower ratio when looking at variation between populations. And then the lowest ratios of all are between cerevisiae and its sibling species, Saccharomyces paradoxus. And of course, this is the simplest explanation is that this is just deleterious mutations that are present in the population that accounts for this high ratio within groups and potentially also accounts for the slightly higher ratios between groups compared to the differences between species. However, there is an alternative explanation involving positive selection, and that is that deleterious SNPs are very likely, since they have to occur, accounting for at least some of this elevated ratio of non-synonymous to synonymous variation within the groups, but that local adaptation has increased the rate of non-synonymous to synonymous variation between these different groups. And if this occurs, it could obscure a signal of positive selection of adaptive evolution between species, because if these numbers are elevated due to local adaptation, then you would detect no elevation of adaptive evolution between species. Local adaptation is just one form of balancing selection, and Wright and Andolfato pointed out that this could be something that was going on particularly in species with strong population structure, such as Saccharomyces cerevisiae and also Arabidopsis. Both of these species also show no evidence of positive selection between species, and so maybe this population structure facilitating local adaptation or some form of balancing selection could be one reason why a signal of positive selection between species was being obscured. And so to look into this in yeast, we took a look at 5 genes that were identified in 2 different studies as showing an exceptionally high rate of non-synonymous variation within species. So from the McDonnell Kreitman test, there's an excess of non-synonymous variation across the species as a whole. And then there was new, more complete genome sequence data available, and we used this and recalculated the number of non-synonymous polymorphism, non-synonymous and synonymous polymorphisms, non-synonymous and synonymous differences between species. And for all 5 of these genes, the McDonnell Kreitman test is significant, and the neutrality index for all of these is above 1, which means that there's an excess of non-synonymous SNPs within the species. As a control, we randomly selected 21 genes and tabulated non-synonymous and synonymous variation within and between species. And this also has a neutrality index greater than 1, which indicates an excess of non-synonymous SNPs, but it's much lower than these 5 genes. And so just as sort of a simple comparison, even though these 5 genes have about half as many non-synonymous substitutions between species, they have 50% more non-synonymous SNPs within species. So these are a selected set of outliers in the genome. And so for these 5 genes, there's 2 models to explain this excess of non-synonymous SNPs. They could be due to deleterious SNPs segregating in the population. That's fairly straightforward and simple explanation. Alternatively, they could be due to balancing selection in the form of, say, local adaptation between different ecological niches. These two hypotheses can be distinguished by looking at synonymous variation. Balancing selection is expected to increase the rate of linked neutral variation, whereas purifying selection is expected to decrease the rate of linked neutral variation through background selection. So these are the results. Out of the 5 genes, one of them shows a very noticeable elevated rate of synonymous polymorphism. So on the graph, synonymous polymorphism is on the y-axis and synonymous divergence is on the x-axis. The neutral, the 21 randomly selected genes are shown in black, and the 5 genes with the high rate of non-synonymous variation are shown in red. So one of them is an outlier. The other 4 are not particularly noticeable. One of the randomly selected genes, TRX2, which is a rather short gene, also appears to be an outlier. But for this one gene, ZRT1, it has a significantly elevated rate of synonymous polymorphism as measured by the HKA test, which compares synonymous polymorphism and divergence, regardless of whether you include TRX2 or not. And so looking at ZRT1 more carefully, this is a tree based on variation in ZRT1. And one of the things to point out is that there's sort of 3 groups of strains, but only one of these groups corresponds reasonably well with known population structure across the genome, and that's this group here, which is mostly consisting of wine European strains. The other 2 groups do not, as best we can tell, correspond to global patterns of population structure. Another thing to point out is that many of the non-synonymous SNPs can be uniquely placed on this tree and occur on these internal lineages. So in our model purifying selection, deleterious SNPs should be rare in the population and should occur on the tips of the tree. Just to give an appreciation of the amount of diversity in ZRT1 compared to the other genes, this is a tree from the concatenated set of control genes, and it's drawn to the same scale. So there's a tremendous amount of variation in ZRT1 compared to other genes. So this is supporting that there's balancing selection is acting on ZRT1 by the high rate of variation. However, there could be selection on genes adjacent to ZRT1, and that could influence neutral variation within ZRT1. So to look at this, we plotted a sliding window of polymorphism and divergence, and polymorphism is shown in black, and you can see the peak of polymorphism occurs directly over ZRT1. On either side, you can see that there's much lower rates of synonymous variation. Both of these genes, MNT2 and FCF1, are not significant by the HKA test compared to the 21 set of control genes. ADH4 is significant, although this appears to be due not to really an elevated rate of synonymous polymorphism, but more because its rate of divergence is exceptionally low. It's got the divergence shown by the dashed line drops off right around ADH4. So what is ZRT1? It's a high affinity zinc transporter. It's induced in its expression about 30-fold under zinc limiting conditions. It's also post-transcriptionally regulated so that when zinc is added back into the media, it's moved away from the membrane and back into the cytoplasm of the cell. Loss of ZRT1 function causes reduced or no growth rate under zinc-limiting conditions, and through interesting but probably an indirect mechanism, loss of ZRT1 also destabilizes microsatellite repeats. And so to look at functional differences between different ZRT1 alleles, we knocked out ZRT1 and found that it caused a fairly dramatic change in growth under zinc-limiting conditions. So here's wild type, here's a ZRT1 knockout. And then we took 3 different ZRT1 alleles from the 3 different groupings of strains that we found. Each of these is distinguished by multiple non-synonymous changes. And we put them back into this ZRT1 deletion strain, and all 3 of them rescue to 100%, at least under the conditions that we were able to measure. So all of them are functionally equivalent and show no differences from one another. Okay, so going back to the original question, does balancing selection obscure evidence of positive selection in yeast? One of the genes that we found, this appears to be the case, in that there's evidence for balancing selection, and this is causing an increase in the number of common non-synonymous synapses, and this will obscure evidence for selection. So just as sort of a rough comparison, if you look at the excess of non-synonymous SNPs in ZRT1, there's 46. In the entire control set of 21 genes, the excess of non-synonymous SNPs is 44. And so I guess the question is, how prevalent is balancing selection? This is hard to answer because balancing selection might not always have a pattern as obvious as that found in ZRT1. In some cases it could be weaker, but it could be acting in many genes and increasing the number of common non-synonymous SNPs. So just to get a little bit of a look into this, if you just plot across all of the yeast genes, the excess of non-synonymous SNPs, there's really no relationship with rates of synonymous polymorphism. And we're not looking at all of the known covariates that influence this relationship, but at least at first glance, there doesn't appear to be some signal that would, make us take a more detailed look at balancing selection across the yeast genome. And so to come back to this data, which is from the Macdonald Kreitman test applied to multiple species, I think that there are people that believe that positive selection is rampant, and the explanation for this data is that the species that don't show evidence for positive selection, the deleterious mutations obscure the signal of positive selection. And on the other side, there's people that believe that this evidence of positive selection is not solid and depends on changes in constraint. And I guess the thing that I would end on is the fact that which I mentioned earlier, which is that Hill-Robertson interference is expected to influence the efficacy of selection, and it's going to depend on the frequency of selected sites in the genome and how much variation is present in our genome. And so I don't think that there's a good explanation yet for this data, but perhaps if we had quantitative predictions for the effects of Hill-Robertson interference on the Macdonald Kreitman test, there would be some further resolution of these types of data. And so this work was done by Betsy Engel, who just defended her thesis last week. And if there's any questions, I'd be glad to take them. Thank you.